Right, yes, there is a very interesting lesson in store for you today. And before we start, do you remember what we learned yesterday? Let me give you a little bit of a hint and get it out of you. This morning, I had sandwich for breakfast, right? I ate a sandwich for breakfast. Now, who can tell me how this sandwich traveled into my body and reached my stomach for digestion? What was the first organ that had my sandwich? Joshua?
The mouth.
Very good. I bit the sandwich and chewed it in my mouth, and it became a bolus before I swallowed it. And then when I swallowed it, where did it go?
The esophagus.
Right, it went into the esophagus. It went through the esophagus in a particular movement. We have a name for this movement. What is this movement called?
Peristalsis.
Wonderful students, you remember so well. Yes, it went through the esophagus through a movement called the peristalsis, and then it reached at my stomach. Then of course you know the stomach has all the digestive enzymes that digested my sandwich until it was absorbed into my body, right? Today we are going to learn a different digestive system, something different from the human, yet it's still the same. So you'll be wondering, what do I mean, different yet the same? Yeah, every living thing has a digestive system. We can leave the plants out of it, the animals I mean. Now, when they consume food, it is going to undergo digestion. However, the systems are a little different. So today, we are going to learn the digestive system of something else. So sit back and enjoy. Today, you're going to learn the digestive systems of the rodent and the ruminant in a very different manner. Not with just the PowerPoint or the textbook, but with an innovative model. Ponzi, the floor is yours.
Now let's look at the models. When the rodents or rats take in their food, the food will first travel into the esophagus. Then later, the food will travel, and next part it will go to the second organ, which is the stomach. Okay, as the digestive system in human, the food will travel to the small intestine. Here is where the difference. In rodents, they have a large cecum and appendix, which have large community of bacteria and protozoa, which produce cellulase. Cellulase is important for the rodents to break down cellulose into glucose. Lastly, the food will be passed out as first batch feces. The first batch feces is watery and soft. But for the rodents, they will consume again the feces and go through the alimentary canal again and pass out as a second batch of feces, which is dry and hard. Now let's look at the light. The light shows the path where the food is consumed into the esophagus, to the stomach, and to the small intestine, but there is no absorption occur. In the cecum and appendix, here is where the important part of organs happen in digestive system of the rodents. Here in the cecum and appendix, the most important part where the cellulose will be digested by the cellulase produced by the bacteria and protozoa here to break down into glucose. But there is no absorption occur because this happen in the large intestine. So the first batch feces will be watery and soft. In the second round of the consuming, when they take in the feces, you will see the food will travel into the same alimentary canal, but into the small intestine, the glucose, which has been broken down, will be absorption will occur. And the second batch of feces will be dry and hard. Understand of this part? Now, when I take down this diagram, what can you see here? What diagram can you see here? Yes, this is a cow, which is a ruminants. Ruminants have a very different digestive system compared to the rodents. Firstly, the ruminants, or which is a cow, have one stomach with 4 chambers. The first 2 chambers here is called rumen and reticulum, as in the rodents. In these 2 chambers, you will see large community of bacteria and protozoa, which produce cellulase to digest cellulose into glucose. But this is not happen in digestive system of human. The third chamber is called omasum, where water is absorbed. The last chamber is called abomasum. This is a true stomach. It's said to be a true stomach because it contains all the digestive enzymes to digest the food. Now, look at the chasing light. The chasing light, first set, you will see when the cow consumes in the food, it will first go into the first chamber, which is a rumen and reticulum. Here is where you will store the food, while the bacteria and protozoa will produce cellulase to break down the cellulose into glucose. There is a different process which occurs only in ruminants. There is regurgitation, means the food will be vomited here into the mouth to re-chew again, bit by bit, before it will be swallowed back into the next third chamber, which is abomasum and omasum and abomasum. They do not go into the rumen and reticulum anymore. After digestion here, the food will be absorbed in small intestine as the normal and go into the large intestine. After that, the undigested one will be excreted out as feces. Look at the yellow path. This path is quite similar when I put on the diagram of a rodent. Look at the yellow path. What can you see? Can you see the similarities between the rodents and the ruminants? Actually, they are the same. Can you see? They are the same. The canals are the same. So you just need to remember the extra dit, which is happen only in the ruminants and not in rodents. Now let's see whether you can answer the question given in the worksheets. Okay, now all of you have the worksheets. The student on my left, please come to the front. I would like you to do some self-learning. Please come to the front. Some of you will control the lights. One of you will explain, and the others help in the labeling. See your label. This is not related. Who is this one? This one. Wait, I have a few here. So you have to pretending answering the question with the worksheets given.
Okay. First, food is consumed and then it passes through the esophagus. And then it goes through the stomach and then the small intestine. After the small intestine, food enters an area called the enlarged cecum and the appendix, which contains bacteria and protozoas. These bacteria and protozoas will produce cellulase, which helps break down cellulose into glucose. After that, the food is passed down as the first batch of feces through the large intestine. And then after that, this first batch of feces is wet and soft. The first batch of feces is then consumed again, and then it passes through the alimentary canal again. And then it's passed out as the second batch of feces. The second batch of feces is hard and dry.
First food enters through the esophagus and moves to the rumen and reticulum. There, bacteria and protozoa produces cellulase to break down cellulose into glucose. The food is then regurgitated out bit by bit into the mouth and chewed thoroughly before being reswallowed, and it enters into the omasum, where water is absorbed, then to the abomasum, where digestive enzymes digest the proteins. Then the food moves to the small intestine, where absorption occurs, and the large intestine. And finally, waste products are excreted out as feces.
All right, dear students. So you had an interesting lesson on the digestive systems of the rodent and the ruminant. So let me just go through it with you very quickly to show you the differences and the similarities. It's not very difficult because it is very similar. The humans, the rodents and the ruminant actually share a common digestive system. The only difference is we have something extra for the ruminant. Let's look at the similarity. After regurgitation, you have this tract for the ruminant. If you were to look at it, it is the same as the tract for the rodent. You can see the yellow lights. They are taking the similar path. Right. So if I were to put a human figure, of course it can't stand, probably lying down from the head going down to the anus, this would be the path as well. So it is like 3 in 1. You have the human, the rodent and the ruminant sharing the same alimentary tract. The only difference would be when we have the ruminant's first stage of digestion, that is before regurgitation. You have the food, we have the food going into the 2 chambers and then coming back to the mouth for rechewing, and then it follows the similar path as the rodent or the human. Is that clear? Yes. I'm sure, because I can see the smiles on your faces. You seemed very enlightened. So definitely you understood, and all the best for the coming exam. Thank you, class.
Thank you, sir.
God bless you. Have a blessed week. Right. This innovative project that the biology teacher did was quite a hit because it was something different from what we usually use in the classroom to teach. Normally, most teachers would resort to the textbooks, and now with the software, we do have these models in our computers, the software where it shows the alimentary canals and things like that. Students are very exposed to all these, and they even can assess this from their home through the internet. But when we have a model with these lights blinking and things like that, it seemed to have caught their interest, and they are more captivated when they are interested, when they are in the classroom, especially when they get to go forward, switch on the lights and then see the flickering lights. They understand the whole subject better, and not only that, it's like a hands-on experience for them. And of course, many other subjects, they were asking if this could be applied for other subjects as well. Yes, it could be, life cycles of a butterfly, or even processes and procedures for an English lesson, BM lesson, we can use them all. But the problem would be if we were to overuse such a project, then it will become a common thing again. To make it different, then it should be like one and the only. Then it gets the interest of the students. Slowly we can develop this for other subjects, but later it will become like it's the light. So until then, it is definitely an innovative project which is successful. Thank you.
One of the objectives of this model is to make the digestive system of rodents and ruminants more clearer and easier for the student to memorize. Furthermore, when the diagram of the rodents is superimposed on the ruminants, straight away the student can see the similarities and the difference between these two systems. Furthermore, students actually can operate the lights and control the models by themselves so that they can do their own self-learning.
It was interesting because the method used by teacher in this lesson was different than normal. It was also because of the lights in the model that caught my attention and made it more meaningful. It was also easier to understand. But it would be better if the teacher used a bigger model because the students at the back can't seem to see clearly until they were called to the front. Yeah, so what do you think?
I think that the lesson was good. Using innovative methods and ideas to carry out a lesson is way better than just teaching using a textbook. I found the lesson very interesting, and the lights that the teacher used attracted our attention, and it left a clearer impression in our mind. The teacher also superimposed the picture of the rabbit on the cow, and this helped us to remember the similarities between the rodents, the digestive systems of the rodents and the ruminants. I think this was really helpful. So all in all, the lesson was very effective as I was able to retain all the information that I learned today.
Yeah, but if this method is continued use among all the other subjects, then it will make this light model thing more special. So if it's used a lot, it will seem a little bit boring. Yeah, but in conclusion, I really like it.